Hey guys, how's it going? This is Dr. Weaver. No introduction other than the fact that this is going to be a hyper-accelerated review on aerobic cellular respiration and photosynthesis. In aerobic cellular respiration, an organic molecule that is ordered, such as glucose, and contains carbon, is going to be oxidized by a molecule, oxygen, and it is going to be used to create ATP, which is the usable form of energy in a cell. As a byproduct, it's going to make carbon dioxide and water. Because we're taking something ordered and breaking it down and releasing the energy, this is going to be an exothermic reaction. Delta G is going to be negative, and the potential and overall energy state of the molecule glucose is going to be decreased, okay, in order to produce the simpler molecules, carbon dioxide and water. If we look at this mitochondria, there's three main parts of the mitochondria that we should be familiar with. This is the outer membrane. There's the inner membrane, which has these folds called cristae to increase surface area, and on the very inside, which is filled with enzymes, this is called the matrix. The three steps of aerobic cellular respiration is the glycolysis, the Krebs cycle, and the electron transport chain. Step number one, glycolysis, really is not part of the aerobic part, but of course it's still needed because oxygen is not needed. It's in the cell membrane, no oxygen is needed, and what happens is glucose is going to be broken down into two molecules of pyruvate. Pyruvate each are 3 carbon molecules. In process, it's going to produce a small number of ATP by substrate level phosphorylation. Also, the electrons that come off of glucose are going to be captured and they're going to be added onto a molecule called NAD. NAD will be reduced to become NADH. A key point here is that if it seems confusing where the electrons are, always look at this hydrogen. The hydrogen is going to bring with it the electrons, so follow the hydrogen, you're following the electron. The second step is Krebs cycle. Krebs cycle is when these pyruvates are going to be converted to acetyl-CoA. You need oxygen for this. Carbon dioxide is produced as a result. Acetyl-CoA is a 2-carbon molecule. It can then enter the matrix of the mitochondria. In the matrix of the mitochondria is where a little bit more ATP is made, also by substrate level phosphorylation, and it also produces the molecule NADH because it captures some more electrons. Also, a molecule called FAD, which is similar to NAD, is going to capture more electrons and become FADH2. Now, FADH2 and NADH, along with the NADH in glucose, are going to yield the most energy in the next step, the electron transport chain. The electron transport chain requires its unique slide, so I can zoom in on the inner membrane of the mitochondria right here. This is perhaps on one of the folds of these cristae. This is the membrane. So if on the top, this is going to be the intermembrane space. The intermembrane space is the space in between the inner membrane and the outer membrane. On the bottom side of this membrane is the matrix. That's the inside of the mitochondria. Here come the NADH and the FADH2s. They're going to drop off their electrons. The energy from the electrons are actually going to provide the force needed to take these hydrogens and have them accumulate on the intermembrane space. When these hydrogen ions accumulate on the intermembrane space, we have a high concentration. Now, by diffusion, they're going to go from high to low. They travel through this enzyme called ATP synthase. ATP synthase is a molecule that is going to convert this energy of the diffusion as it's flowing through, kind of like a windmill or a watermill, to create ATP from the building blocks ADP and the phosphate group. Okay, this is going to flow into the matrix. Now, one common question that occurs a lot is going to be, what is the final electron acceptor in this electron transport chain? And that is going to be oxygen. Oxygen loves electrons. It will take the electron and it is going to combine with the hydrogen that comes down, not in this ratio, but it will indeed make water. That's where water comes in from the equation. So what are the terms associated with this? Well, the osmosis of this ion or chemical that flows through is a process called chemiosmosis, and the process that makes this ATP is actually going to be called not substrate level phosphorylation as in the glycolysis in the Krebs cycle, but because these are oxidative reduction reactions, this is going to be known as oxidative phosphorylation. Now, when oxygen is not present, some organisms are adapted to do anaerobic fermentation. Anaerobic fermentation is when it primarily focuses on glycolysis. But in order to continue glycolysis, you need to continue taking electrons away from, such as glucose. So what happens is the two pyruvates that are produced as a result of glycolysis can actually be modified to free NAD again. So the NADH has the electrons. If all of the NADs are tied up with the electrons, pretty soon you're not going to have enough NADs to continue glycolysis by to take the electrons from glucose and oxidize it. So what happens is pyruvate is either going to get converted to alcohol and CO2 in the case of alcohol fermentation. This is going to be the case of certain organisms such as yeast or some bacteria, in which are going to make ethyl alcohol, and which is a 2-carbon molecule, and carbon dioxide, which is a 1-carbon molecule. This is very useful in the beer and wine industry, and also making bread, which is where the CO2 bubbles come from for the rising of the bread. Lactic acid is the other method that some organisms use. A lot of times people think of the muscles building up lactic acid when a person is in oxygen debt. Okay, this is a 3-carbon molecule, so there is no carbon being released, so that means there is no carbon dioxide. This just gets conserved as 3 carbons as well. Also, in the industry of yogurt, certain bacteria are going to undergo lactic acid fermentation. Okay, on to photosynthesis. Photosynthesis is going to be the process where it's going to take the energy from the light, and it's actually going to store it in the form of an organic molecule, like a sugar. In order to do this, it's going to take the electrons from water. When the electrons are taken from water, they're going to be used to make ATP. This ATP is going to be used with the electrons to link together carbon dioxide in order to help produce sugar. This, because we're creating something ordered, it is going to be endergonic. It is going to be absorbing the sun's energy. Delta G is going to be positive because we are ordering it together in something more complex. And of course, if we're increasing the energy state and we're storing the energy, we are increasing the potential energy. Here's a chloroplast right here. Here's a big outer membrane of the chloroplast. Now, the functional unit of the chloroplast of capturing the light is going to be these thylakoids. Now, the thylakoids are actually going to be stacked in these grana, singular granum, and each thylakoid has a membrane around it. If I were to draw an analogy to a mitochondria, I would say that the inner membrane of the mitochondria, where the electron transport chain takes place, is very similar to the membrane of the thylakoid. But instead of having folds like the mitochondria, we actually take what may be a larger structure and break it into a bunch of little structures, flattened pancakes, to increase the surface area for the electron transport chain to take place at the thylakoid membrane. Now, the way that this works is light is going to come in, used to break apart water into oxygen, and then what happens is the oxygen actually gets released. The electrons are going to be used to generate ATP. Now, the ATP, as well as the electrons being captured on a molecule similar to NAD. NADP is in photosynthesis. You could always remember the P for because it kind of like stands for photosynthesis, even though it doesn't, but that's how you can remember it. NADPH is going to carry the electron, and it's going to bring it to the stroma. The stroma is the liquid part of the chloroplast. I would say it's kind of like the matrix of the mitochondria. The liquid part of the mitochondria that has the enzymes is the matrix, while the part with the enzymes that's liquid in the chloroplast is the stroma. Here the Calvin cycle goes on. Calvin cycle is the carbon fixation process, where carbon dioxide is going to come in, get fixed to make a sugar. Okay, in the thylakoid, we have 2 reaction centers, photosystem 2 and photosystem 1, also nicknamed with the antenna molecules that are going to be capturing the light. These are actually going to be embedded in the thylakoid membrane. So here's what happens. Light comes in, the electrons in water to bring them to a higher energy state. As a process, the oxygen from the actual water molecules get released. Don't forget what I said before. Follow the hydrogen, you're following the electron. So here comes the electrons. With that hydrogen, the electrons are going to be coming down the electron transport chain. The hydrogen ions are going to be setting up in a concentration gradient in the actual thylakoid membrane. The hydrogen ions are actually going to be inside the thylakoid and flowing out by chemiosmosis to make ATP. Now, because this is driven by the light, we call it photophosphorylation. So by photophosphorylation, it makes ATP. In the next photosystem, light comes in again. It's going to excite the electrons, and it's going to take one of 2 paths. It's either going to go back to photosystem number 2 in order to be re-excited to make more ATP. This is a cyclic portion of the cycle. We could call it cyclic photophosphorylation, where it keeps making ATP. The other road is that it is going to go on the molecule I said before, NADP, in order to reduce NADP to become NADPH. This is going to go to the Calvin cycle. So one common theme is that the products of the light reaction, which is right here in the thylakoid, are used to drive the process in the Calvin cycle. Okay, so up next, the absorbance of light is going to be something that you have to be familiar with. If we look at the absorption of light and we look at two pigments, chlorophyll A and chlorophyll B, we can actually graph the absorbance of how much energy it absorbs based on comparing it to the wavelength. So you can see in the purpley blue range, we have chlorophyll A and chlorophyll B are going to absorb the most, and also they're going to absorb the most energy at a wavelength in the red range. Now, the evolutionary significance of having two different types of chlorophyll, and actually there are other supporting pigments that help even broaden the range, is that it gives more of a wide range of wavelengths that it can absorb. And what color are plants? They're green. If they're green, that means they're not absorbing green, that means they're reflecting green. So a very famous experiment was done where they took a piece of algae. So this green strip is a piece of a filament of algae, and what they did was they took a prism where it separated the light into the various wavelengths of light, and they allowed this aerobic bacteria to be in the presence on a petri dish perhaps, and they were able to see where the bacteria go. The bacteria could actually move. Now, the bacteria like oxygen. Photosynthesis produces oxygen. The actual bacteria actually went to the regions of light in which corresponded directly to what wavelengths of light were absorbed by photosynthesis. So what's going on in the stroma? In the stroma, we have carbon dioxide coming in. The enzyme Rubisco is going to fix carbon dioxide with a molecule called RuBP. RuBP is ribulose bisphosphate, and what happens is it is an enzyme that is going to rearrange the molecules, and what happens is ATP comes in from the light reaction. The electrons on NADPH come in from the light reaction. What do you need to energize this is a phosphate group. What do you need to link the carbon dioxide? The electrons are going to form the bonds in these molecules. Well, look right here. We have carbon in, carbon out. G3P is an intermediary molecule for glucose. It's actually 3 carbons. Glucose is 6 carbons. Isn't that interesting that in cell respiration, glycolysis, glucose is broken down into 2 3-carbon molecule pyruvate? Well, here it is linked as a 3-carbon molecule as the building block of glucose. What is going on with these guard cells? Well, guard cells are in charge of actually, when they're open, gas exchange. Carbon dioxide goes in, and then other gas can leave. And then when they're closed, carbon dioxide cannot come in. Why would they close? A hot, dry day, what's going to happen is that they need to close in order to conserve water. However, this stops the gas exchange. This is bad because what happens is oxygen, which is inside of the plant as a byproduct of photosynthesis, will compete with carbon dioxide for binding to Rubisco. Now that carbon dioxide is no longer in the picture, it will now bind to Rubisco. It will try to continue to put out glucose. Well, if there's no carbon in and you're getting carbon out, it actually starts deteriorating the system. So what's going to happen is this is called photorespiration. Photorespiration is bad for the cell. So there are actually 2 adaptations for certain plants that to deal with the idea of photorespiration. Now, a plant that does not have these adaptations are called a C3 plant. Plants that have adaptations are the C4 plant and the CAM plant. What they both do is they both make a 4-carbon acid, which is a stored version of the carbon, and then there's carbon dioxide not available. It will actually break down this acid and release carbon dioxide inside the leaf. So this way it can be used for photosynthesis. Now the difference between CAM plants and C4 plants is CAM plants are going to bank CO2 at night when the stomates are open. This is an organism such as a cactus in a desert. A desert has hot days and cold nights. That means that at nighttime the stomates can open because it's cool, brings in the carbon dioxide and stores it as the 4-carbon acid, and then what's going to happen is during the day when it's closed, it's going to break it down into CO2 for photosynthesis. Now with C4 plants are going to be doing different compartments. There's the mesophyll cells and the bundle sheath cells. And the bundle sheath cells is able to take in carbon dioxide and store it in there, and then this way bring it to the mesophyll cells to do photosynthesis. So it's actually spatial separation rather than time. rather than the time of day. Well, that's it. That's review of photosynthesis and respiration. I hope you guys got a lot out of it, and make sure that you look in your Cliff's book or whatever review book that you're looking at and to dig more deep.